Hello, good afternoon. Just realised I haven't tweeted out a link today, but I think there's enough of you out there who come along every day that we should be okay. Okay, hello everybody. Welcome to Wednesday. The days really do blur into one. It's Wednesday, and it's day number 21. And hey, look, there are 21 of you. Oh, no, 20 of you. Okay. Okay, right. So, yes, day number 21. And what are we going to do today? We're going to start to talk about R. So, I'm quite excited about this. Just for full disclosure, this will be the first time I've properly taught R to anyone. I've been involved in lots of courses where R has been taught, but I've not been the one doing the teaching. So be brave, everybody. We'll get through it together, and I'm sure we'll be fine. What we're going to do first. So we're going to talk about R, obviously, and that's kind of a lot of what we're going to do today. But what we're going to start by doing is just quickly taking a look at the output of salmon from yesterday. Yesterday we kind of finished up at the end of the hour. We had written our script that was designed to run R, and we had designed to run salmon. Sorry, I'm trying to do three things at once here. We'd written our script that was the whole point of the script was to run salmon over our 12 samples. And if you remember yesterday, we had there was a bit of kind of work to do to get from this list of 48 SRA accession numbers to a set of 12 samples, each represented by 4 FASTQ files that we could use for quantification using salmon. So we wrote all of that stuff into our script, and then at the end of the hour I started the script running with a promise that we would come back today and check that we've got the output from salmon for those 12 samples. So okay, my VM isn't currently running, so I'm going to have to boot my VM up. I'm going to start VirtualBox and boot up my VM. So let's do that first of all. I wasn't necessarily planning on running the VM very much anymore, which is why mine was shut down. But we do need to have a check of our salmon output before we go any further. Let's not get too carried away. So I'm just booting up my VM, and then we'll get a terminal and we'll have a quick look at the outputs from yesterday. Just another quick note on yesterday's work, actually, before we go any further. I did put it. It was in the show notes for yesterday, and it's also. I think I tweeted a link out to the GitHub because I did put the script that we wrote yesterday is in GitHub, so it is available in GitHub in the week 5 directory. So you can see that script that we wrote yesterday there. And also I put up, as promised, the 256 SHA-256 sums of all of my 48 FASTQ files. So you've got a list of those there in that text file. So you've got the hash of the file followed by the name of the file that it's a hash of. And then I also put up the results of ls-l in my week 5 directory, just to show you the size of all of the FASTQ files. And so this gives you the number of bytes in each of these files. And for most purposes, checking the number of bytes in your files is the same as the number of bytes in mine should be sufficient. because they should be directly comparable. But if that's problematic, and actually sometimes the hashes are easier to check the SHA-256s just to check that we've got the same data, that we've run salmon over the same data. So I just wanted to draw your attention to the fact that that data is available on GitHub. Okay, so let's have a look at the results of yesterday's work. So in our share directory and in week 5, that's where we ran salmon. And what we should hopefully find is that we've got, for each of our samples, so that's for each of our geo sample IDs, so the GSM accessions, we have a directory, and that's the output of salmon. Lives in those directories. So I should here have 12 directories that begin with GSM. Okay, so as we've been doing for the last couple of weeks, let's just check that we have. That's not what I expected to see. Oh, that's because it's listing all of the contents of the directories as well. Okay, fine. Let's just count them. There's only 12. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Okay, yeah, fine. I've got 12 of those directories. And in each of those directories, we should find a quant.sf file. So actually, that's how we can check it. GSM start / quant.sf. There should be 12 of those. And that's one per sample, and there is 12 of those. There you go. So that is the output from salmon, the primary output. There's lots of files that come out of salmon, and if we go into the first one of these files to have directories to have a look, 7.8 is the first one, and list that, you'll see that there's a number of directories and files that are the output of salmon. Things like the command that we pass to salmon, which is expressed in this JSON format, which is a JavaScript data format, but it's a fairly convenient short way of expressing nested data structures like this. So that's the parameters that we passed to salmon, among other things. There's also things about lib. So lib format counts is how it determines. If you remember, we said to do this auto detection of the library type. Well, the lib format counts is what it uses to generate that. So you've got all these different types of library, and each one of these initializations stands for something. So the ones which I know are inward stranded reverse and outward stranded reverse. So they're kind of major library types. Certainly inward stranded reverse is for paired end libraries. You expect most paired end libraries in this day and age to be inward stranded reverse, because that's what the Illumina TrueSeq protocol gives you. We've got a single end library, so we can't determine whether our library is inward or outward. That is whether the reads are pointing towards whether your pair of reads are pointing towards each other or away from each other. Inward if they're pointing towards each other, outward if they're pointing away. But we can determine if it's stranded. So all of our reads should map to the template strand of the corresponding to the gene. So should map in the gene, and they should map to the template strand because it's a stranded library. And they should, in general, they're mapping in the reverse direction, so they're pointing in the opposite direction to the gene. So it's stranded, and it's stranded reverse. That's the thing that's telling you that they map to the template strand. So almost all of the reads in this particular experiment conform to that configuration, and just a tiny fraction of them are stranded forward, so going the other way to the stranded reverse, so mapping to the coding strand. So that's good to see, because it just means that the library prep here is really, really consistent. Pretty much all of the reads conform to the format that we expect. So the only other thing I want to talk about is the quant.sf file. So let's have a quick look at that. So this is the primary output of salmon, and this is for each transcript in your transcriptome index, that is the index that we built on Monday using Salmon index, for every transcript in that index, we have a quantification. So we have four numbers. Yes, four numbers. The length, what's called the effective length. That's because depending on the fragment size, you can't get fragments that map all the way to the end of genes very well. So you reduce the length by in relation to the fragment length. So yes, length, fragment length. Then TPM, that's a normalized measure of the number of reads that map. So normalized by the size of the library and the length of the transcript. TPM stands for transcripts per million. And then number of reads. So this is your raw number of the number of reads that map to this transcript. And you will notice that that count is fractional, and that's because of the way that this is calculated. Strandedness is known without mapping, Graham, in the comments, because Salmon sort of is mapping. It's finding a sequence-based match for your read in your transcriptome. So although it's not producing a mapping, strictly speaking, so it's not producing an alignment, it still is looking for the best match between the sequence of your read and the sequence of your transcriptome. So it is doing some sort of mapping procedure, and that tells you that in the process of doing that, it can determine whether it's a sense match or an anti-sense match. So it can figure out whether your prep is stranded or not from that. Okay, so that's the output of Salmon, and this we will come back to later. Bear in mind that multiple transcripts here belong to individual genes. So it may be, I can't remember exactly, it may be that the first two transcripts here both belong to the same gene, and actually neither of them is expressed. We get 0 reads for both of them. But what we'll be doing in R when we get that far is summing up the transcripts that belong to individual genes. We'll be summing up their expression to give us a gene level measure for the expression of a given gene. Okay, so that's the output of Salmon. What we haven't done at this stage, and it may be that it's worth coming back to this at some point in the future and reconsidering this, we've not considered the expression of the viral genes at all. So we know that there's that half of these samples have been infected with the COVID-19, but with the virus that causes COVID-19. So there should be viral expression, at least in those samples. At this point, our transcriptome is just the human transcriptome. We're not considering the viral expression at all. We might take a quick look. So one of the outputs in the logs will be the mapping rate. So in the logs, in this log, we should be able to find an entry about the mapping, and you could maybe, there you go, mapping rate 88.97%. You could maybe use differing mapping rates to tell you something about viral expression, because you would assume that in viral samples a certain amount of the reads are going to go on mapping to the viral genome, or the viral transcriptome, so we might see a reduction in the mapping rates of the human transcriptome. We can maybe take a look at that later, but right now I want to talk about R, and I want to get us moving with R, because that is kind of the point of today. I do realise I usually spend 15 minutes talking about yesterday before moving on to today, but that's the nature of the beast. Okay, so R. So R is a statistical programming framework. So it's written, it evolved from an earlier programming language called, imaginatively called S. It's written by statisticians, and it's intended for doing statistics. So it's very, very good at working with numerical data. And it has evolved, especially in the last 7 or 8 years, to be a much more general purpose, what is now referred to as data science programming language. Quite how you do science without data, I'm not entirely sure. But yes, so it's evolved from this kind of statistical framework for applying statistics to tables of numbers and lists of numbers and so on and so forth, to being a much more kind of general purpose, just working with data language. What we want it for is obviously for doing bioinformatics, but essentially what we use R for in bioinformatics is mostly statistics. It's almost entirely applying appropriate statistics to data. And it's a very good language for doing that. It's got the backing of decades of statistical research. And most importantly, for the things that we care about over the next couple of weeks, or the remainder of this course is it's got bioconductor. So bioconductor is again a reasonably long in the tooth project. It's been around for a long time, and it's a collection of packages for working with bioinformatics data. So it's now, I think, well over 1,000 packages aimed at working with lots and lots of different types of biological data. And I refer to it as an enormous repository of other people's intellectual effort. So the chances are, if you want to analyse biological data in some meaningful way, there is almost certainly a package for doing that analysis in some way, at least, available to you on Bioconductor already. Somebody has already spent the development effort to come up with a way of doing that analysis. The way that you put those packages together, the way that you build your analysis together may be novel, but the chances are that somebody has already analysed data in the same way, and the chances are they have a package on Bioconductor to help you do the same thing. So Bioconductor is a massive resource. It's a huge resource for doing bioinformatics, and particularly for the kind of statistical analysis that we want to focus on with our RNA-Seq data here. There's lots and lots of options for analysing this kind of data on Bioconductor. So that's kind of my motivation for starting us off with R now, is we have some numbers generated by salmon, and we want to analyse those numbers in the best way, and R provides us with that best way. On top of that, R is a bona fide Turing-complete programming language, just like Bash is. It's got its quirks. It's quite archaic in lots of ways, but it's an extensible programming environment. So we can add packages to it. We can add functionality to it with packages, which is the whole idea behind Bioconductor, really. And we can use those packages to make R into a much more modern framework than would otherwise be the case. So although the language itself has some ticks, actually the ins and outs of it are really enhanced by the fact that it's extensible, and there's a huge amount of packages available which can turn it into a really robust modern programming language. Okay, so what we're going to do first of all is we're going to install R, because I don't want to demonstrate anything until we've got R up and running for our own purposes on our machines. So the first thing that we're going to do is we're going to download and install R. Now, On my laptop, I already have R downloaded and installed because I use R pretty much every day in my regular work. So it would be remiss of me not to have an installation of R. But nevertheless, I'm going to walk through this procedure anyway, and it may well be that I know that there is very imminent. There is a very significant release of R due. So at the moment, the version of R is, I think, something like 3.6.3 or something like that. R version 4 is just about to be released, I think, in the next week or so. So I'm aware that I'm doing this at a really bad time. But nevertheless, it may be that there's a more recent version of R that's been released since I last installed R on my machine. So let's begin with installing R. So okay, so R is distributed from the R project website, which is r-project.org. Okay, so that's the R website. There you go. So R version 4 pre-release versions will appear starting March 24th, scheduled to Friday. Final release is scheduled for Friday. So yes, I knew we were doing this at a relatively bad time. We are going, for the purposes of right now, we are going to download R 3.6.3. We might look at upgrading R to version 4 in a couple of weeks' time. New major releases, it's never sensible to upgrade or to download them on the first day of release, or to upgrade straight away, because there's usually some stuff to be worked out of them. So we're not going to install R version 4 straight away. For our immediate purposes, we're going to install R version 3.6.3, which was released at the end of February. So this is the version of R that we want. The second link down in the news. So we're going to click on that. So this is the source. So that's not what we want to download. So I take that back. Let's not click on that. If we want to download R, let's click on the Download R button here. Okay, so download R, and then pick one of these mirrors. One that's geographically close to you will probably be fine. The top one, the R project main mirror, that will also be fine. That will work fairly quickly. So there you go. So now we get the option. So download R for Linux, for Mac, or for Windows. So pick the appropriate distribution to download a compiled version of R for your platform. So I'm on a Mac, so I'm going to download the Mac version. Obviously, if you're on Windows, download the Windows version. I should emphasize at this point, I'm doing this on my host. I'm doing this on my laptop. I'm not doing it on my virtual machine. Okay. You could install R on your virtual machine. There's nothing to stop you installing R on your virtual machine. And there's some reasons why that might be desirable, but in general, I think it makes more sense. And the reason why I set up the file share in the first place at the beginning of the course was because my intention was always to install R on the host, to install R on the machine that's sat on your desk rather than on the virtual machine, because then you get all of the resources of your main machine for R. And because you've got this shared file set up, you can still access the files that we created in the VM. And then later on, if we go back to the VM can also access the files that you're going to write out to that same shared directory when we're working with R. Okay, so we're going to download R for the appropriate platform. So I'm going to click download R for Mac because that's my platform, and I'm going to pick. So that's dropped me down the page to the right place, and I'm going to pick r363.pkg. So that's my package that I want to download. Okay, so I'm going to start that downloading. If you were on a Windows machine, then click the download R for Windows, and then pick the base distribution. Okay, so download that the base distribution of R for Windows. and there you go, that R363 for Windows. So that's what you want to download. And then follow the installation instructions. So for Mac, this is relatively easy. I don't have a PC in front of me, so I can't follow through the Windows instructions. But hopefully it should be relatively self-explanatory. I assume this is going to download an MSI or something similar that you can double click and it will start to install stuff. For Mac, we've downloaded a package file, which we can then run once it's downloaded. I'm about halfway done. So this is downloading R, and once we've downloaded and installed this, we are halfway towards achieving a version of R that we can really use in anger. Because I've already got it installed, actually, I'm just going to demonstrate. So I'm just going to open a terminal on my machine, and I'm going to cd. I'll put this. I'll put this in the right place in a second. Okay, so I've got, this is a terminal on my Mac. So I've opened the terminal application on my Mac. If you're on Windows, you can open the command interface on Windows as well, and it's exactly the same thing. I've already installed R on this system, so I have a working installation of R. So if I just type R at the command line, R is in my path. It has a binary on the system, so I can invoke it from the command line, and you can see that it drops me into an R command prompt. So now, instead of being at my Mac Unix command line, I am now at the command line in R, and I can type commands in R, like 1 plus 2. R does simple computation quite happily, so 1 plus 2 equals 3, right? That's fair enough. And it's R that's executing those things for me. It's not my command line interface, not my usual command line interface. So I am in R here, and you can see when it starts up, it tells me the version of R that I've got installed. So I am indeed updating R when I'm downloading this new version, because this is R360 and I'm downloading R363. So this once upon a time, this was the primary interface to R. This was how you worked with R as a programming language, was via this kind of interactive interface, or you could write scripts and you could run them via a binary called RScript, which let you run scripts at the command line. But the thing that really changed R use for me and really revolutionized my use of R and turned it into my everyday programming language rather than Python, which is what I used before, is the availability of another piece of software which is often installed alongside R, called RStudio. And RStudio is what's called an integrated development environment, or an IDE, for writing R code, and it is it was, for me anyway, as a bioinformatician, a truly kind of life-changing piece of software, because it meant that I could escape that sort of clunky command line interface and gives you a much more modern interface to R as a programming language. So the other thing that we're going to download is RStudio. So rstudio.com. So these are the good people that develop it. RStudio is freely available, although it's not free software. It's commercial software, and you can pay for a license, but it is freely available for most people to use. You can pay them for extra support and for various fancy versions, but we just want to download the open source version of RStudio. Okay, so this is what we want to download. So if you go to products and then RStudio under the open source section, this is the thing that we want to download. And we want RStudio desktop. Okay, so not RStudio server, but RStudio desktop. And there will be, if we go down, download RStudio desktop. Apparently they want you to click lots of times. We want the free version. Many clicks, and eventually, and I assume this is doing this relatively intelligently, it's given me a link for downloading it for Mac. So I assume if you visit this page, and you're on a PC, it will give you a link for downloading it on a PC. So I'm going to download RStudio as well. That's the other thing that I want to download. So we'll click on that and we'll get that downloaded too. But we want to install, before we install RStudio, we need to install R. So let's do that first. So I'm going to open the package file that I downloaded, which is my R installation package. And then I'm going to follow through the on-screen instructions to get this installed. So this will install R for me. In this case, it's a newer version than I've got. I've already got R installed, but this is installing a newer version of R for me. Okay, so we'll just continue and continue and continue, and we'll agree to the license, install it for everybody, even though I'm the only user. And there we go. So that's going to install R for me. So away it goes, putting the files in the relevant place, installing me a new version of R over the top of my old version of R, and very nearly done. Shouldn't take very long because it's not a huge installation. So this is just getting to the point of finishing up, and then we should have our installation of R ready to go, and then we just need to worry about RStudio. So there you go, installation was successful. So let's close that, get rid of the package that we downloaded, bring my terminal back up and just show you that by typing R now in my terminal, re-invoking R, you will see that the version has changed in this little introductory text that you get when R starts. So instead of saying 360, it now says 363. So I have successfully upgraded my R installation. Okay, so that's got me the latest version of R until Friday. So get rid of my terminal again. The next thing is to install RStudio. So again, RStudio, their aim is to make this easy. It's like any other, many other applications on the Mac. I've downloaded a disk image, DMG, and this will open up and give me an icon that I can drag into my applications folder. So this is the installation of applications on a Mac. On Windows, I assume that again you will have downloaded some sort of exe or MSI file that will lead you through an installation process that will install RStudio into your programs on your window on your PC. So again, you can see I've already got RStudio installed because I've already used it. It's an older item named RStudio. So once again, I'm probably updating my software on the fly here. It's no surprise to me that I've already got it installed. I knew that, but let's replace it. So let's install our new version of RStudio. And then we'll close this down. And then we can run RStudio. So now that it's installed, I can run the application from my applications folder. I've got a version of it on my dock, which isn't on the bit of the screen that I share. But I'm gonna click the RStudio icon down in my dock. I could equally navigate to my applications folder. If you're on Windows, it will be under your start menu in your program files. You'll be able to track down RStudio in there. And RStudio is our primary interface to R. We're not going to go back to that command line version that I've already shown you. Okay, so I'm gonna open up RStudio. I'm just gonna whisk it off the screen for you as it boots up because it's going to come up with a whole load of my stuff already up in the screen, and I'm gonna make sure that I'm gonna clean up my view before I show you. So here we go. We're gonna close the current project. So I forgot to do this before we started, which is what I really should have done. Okay, it's getting there. Just waiting for it to start a new session. There we go. So let's close these things up as well. Okay, so hopefully in the time that's taken me, some of you will have been able to get R downloaded and installed. If not, it should be relatively straightforward. It's in these people's interests to make it straightforward. Okay, here we go. So, RStudio. I think this is relatively good to go. So here we go. This is RStudio. So when it opens up, you should, I think, have these three sort of panes, these three panels in your view. One of them is the console. So this is, if you look at it, actually, let me just drag my terminal up here like this. You'll see that it's exactly the same as the text that appears in the terminal when you run R at the terminal. So let me just get slightly smaller. There we go. So you can see that we've got this R version 363, and then the date, and then the name of the R release things. This is all the same text here in RStudio as it is here in my terminal, and that's because that's effectively what's happening in this particular pane. Is you've got this is our R terminal in exactly the same interface as we have it at the command line. But we also have some other views. So this pane has some tabs. So we've got an actual terminal. This is a command line terminal on my Mac. So I can type my standard terminal commands here, and it will work exactly like our command line terminal that we've been using for the last few weeks. And also I have this interface to jobs, which we're not going to worry about for now. So we get different tabs in our different, and we've got this arrangement of panes and tabs. So I've got a pane at the top here, which has the environment, the history, and connections. So we don't need to worry too much about these things at the moment. I'll explain these as we go along in future lessons. And then at the bottom here, we've got other things. So files, plots, packages, help, and viewer. So again, a whole range of things in there as well. Okay, so we will explore what all of this means as we go along. But the nice thing about RStudio is that these different panes are all linked up. They all work with each other in sync. So as I do something in one of the panes, if it affects what's going on in the other panes, then the change is reflected in those other places. And again, you'll see what we mean as we go forward. Now, the one thing that I would say about my setup here is obviously in order to show you this, I'm sharing my screen with you. And so I have limited space to get RStudio on the screen. You know, I'm sat in front of a nice 4K monitor, a quarter of which is being shared with you. So this is this is the top left hand quarter of my screen that you're seeing here. And so that's fine, but it doesn't give me a huge amount of real estate. So I am going to have to kind of jiggle about with RStudio a little bit to make sure that the things that I'm doing are visible. But I just wanted to share with you, this is the default layout. And so if I change things as I go, it's only because I want you to be able to see what I'm doing, not because I necessarily would normally set things up like that. I quite like the default layout. It's quite helpful. Okay, so this is the basic layout. These three panes. We actually usually, once you're actually working in RStudio, you usually see a fourth pane, and that is the script pane. So just to demonstrate that, I'm going to create a new script. So I'm going to this is the create new button up here in the top left hand corner of the interface. So I'm going to click that, and I'm going to create a new R script. Okay, so just an empty R script. So a script here is exactly the same in terms of what it is as an entity as our bash scripts have been. So our bash scripts are text files into which we put some special syntax, and they're interpreted by the bash interpreter. So all of those scripts that we've written over the last few weeks, they're just in the end, when you just look on the file system, they're just text files. And exactly the same is true of R scripts. They're just text files. So we're going to create an empty text file, but we're calling it an R script because we're going to put R commands into our R script. And that opens a fourth pane, and by default it's above the console pane. Okay, so the reason that I've done this is because, especially when we really get going in anger, it's relatively good practice to write scripts rather than write commands at the console. And that's because, for the same reason as you would write scripts in Bash, it means that you can rerun those processes easily, that you can come back to those scripts at a later date and see what it is that you did. And also it's kind of pulling a set of commands together into one place, and a set of commands to do a job together in one place. And if you didn't do that, if you just executed commands at the R console, which you absolutely can do, but if that's all you ever did, you wouldn't ever have processes that hang together sensibly. Finding out what you did to do a particular analysis would be quite difficult to do. So typically speaking, we will usually write a script and save our commands in the script. So just to show you the interactivity of the different panes here. So I showed you before that at the console in R, we can do at the very simplest level, we can begin by just running some simple calculations. So 1 plus 2, right? Simple arithmetic. Wonder why I got those. That's interesting. I think I might just, I may well start a new R session. That was an interesting set of warning messages for doing some computation. Let's try again, see if we get the same. If we get the same, then I will reset my R. No, that's fine. Right. So there you go. Simple computation. 1 plus 2 equals 3. Okay. So you type in the computation, you hit enter, and R gives you the result interactively. Okay, so that's your interactive console, and you can absolutely run commands there, and it works just fine. You can do all of your usual things: addition, subtraction. We can raise 2 to the power of 2 by using the caret symbol, or 2 to the power of 3. By using the caret symbol. So we have all of the usual options available to us: 2 times 3, 3 divided by 2. Okay, so that's a good question. Do you need to install developer tools on Mac to work with RStudio? Possibly. This is the problem with me not installing this fresh, is there may be some dependencies. I think Peter's right. I don't think you do, but you may do for some packages later on, but I think you'd probably be okay, to be honest. Right, okay, so yes, so that's simple arithmetic, and you can do it at the console, and you can do it interactively. We can also do it in our script, so we can write out the arithmetic in our script, and when we press enter, that is not executed. It's a line of a script. Okay, so there's nothing about that says that we're going to execute that command. You can execute it by going to the line that it's on and pressing Control and Enter. I think Control and Enter works on a Mac. Yes, it does. Or Command and Enter works as well on a Mac, but Control and Enter. And what Control and Enter does is it sends the command on the current line to the console. Okay, so you can see that in just demonstrating that, I just executed 1 plus 2 3 times in a row because I pressed Control and Enter, then Command and Enter, and then Control and Enter again on the same line. So Control and Enter on a particular line in your script sends that line to your console and executes it at the console. Okay, so that's kind of your simple computation, and we could put in any of those other things that we did before. So 3 divided by 2, 3 multiplied by 2, 3 to the power of 2, 3 minus 2, right? It's all simple computation, and then you can step through it. So every time you control and enter, your cursor goes down a line, so we can step through each of our commands by pressing control and enter repeatedly, and it steps down line by line and executes each of those lines of code. Alternatively, you can highlight a block of commands just by using your mouse or using the arrow keys on the keyboard with shift depressed, and highlight a block of code, and then when you press control and enter, all of those lines get executed one after the other, again in the order they are in the script. Okay, so 1 plus 2, 3 over 2, etc. So all of those lines get executed one by one, again in the order they are in the script. So you can highlight blocks and run blocks of code like that as well. So that's showing you how the script, your script, interacts with your console, and this is true generally speaking. We can also get it to interact with some of the other panes. So we can do variable assignment in R just like we were doing in Bash. So we can set variables using assignment. There are two ways of doing assignment. To keep things simple, because it's what we use in Bash, I'm going to use the equals sign for assignment. So this is assigning the value to the right of the equal sign to the variable on the left. So our variable here is called x, and we're going to assign x the value of 3. Okay, so that's the line of code to create the variable. And when we execute that line of code, so I'm going to press Control and Enter to execute that line of code. So this time, what's happened is this line of code has been executed. So we've got x equals 3, which has been sent to the console, and it's been executed. This time, R gives us no output. So we've set a variable, but R doesn't give us any response. It doesn't tell us what we've done. If we want to see the results, we can do x, and just by typing x, that will print out the variable. If you want to see the results of a command as you run it as well, you can put it in brackets, and I think that will print out the value of the variable as well. But the other thing that's happened, which maybe you might not have recognized, is that x, which is our variable, has been added to what's called our environment. So in our environment, we've picked up the variable x here, and x has been given the value of 3. Okay, so that variable has been defined in our environment, and that means that x is saved. We can come back to x later on. We can use x in our calculations. So we could use x in our arithmetic. So we could do 4 minus x. In fact, let me put it in the script. Let's keep our good practice going. So 4 minus x in our script, and then we can execute that line of our script, and you see the result is 1, and that's because x is 3. Right, so 4 minus 3 is obviously 1. We can change the value of x, so we can set x equal to 2 and rerun, and you can see in our environment the value of x changes. So we now don't have 2 different x's, we've just changed the value of x to 2. And now if we repeat our previous computation, even if we do it on line 7, don't write it out again, but run it on line 7, x's value is 2, and x's value is being respected by that computation. So 4 minus x now gives us the result 2 rather than 1, and that's because we've changed the value of x. And you don't just have to assign x to a simple number. We could assign x to the results of a computation. So you could say, or we want x to be the results of 3 to the power of 2, in which case x becomes 3 squared. So if we do Control X again, x becomes 3 squared, which obviously is 9. So now, if we now rerun 4 minus x, we will get minus 5. Okay, so this is just the beginnings of exploring R. The one other thing which I'm going to do just to demonstrate the full kind of interactivity between all of our different panes here is R has functions, and we'll come back to functions in the future. But let's just do a very simple function to begin with, which is plot. So plot will take the input and produce you a suitable plot from it. So if you give it 2 lists of numbers, then it will plot those 2 lists of numbers against one another in a scatter plot. So if we just give it a list of numbers from 1 to 10, and we define that with the colon operator. So the 1 colon 10 will give me a list of numbers from 1 to 10. And then if we do the same on the other, in the second argument to plot. So here plot is a function, and functions, the arguments to functions are defined using inside brackets. So you do plot, open brackets, then the arguments to plot, and then you close the brackets. Now when we execute this, R is going to run the plot function with those 2 arguments. And the 2 arguments here are the 2 lists of numbers 1 to 10. This will produce a scatter plot with points at 1, 1, 2, 2, 3, 3, 4, 4, and so on. So a diagonal line of points across our plot. And that plot will appear in the plots pane, which by default is in the bottom right down here. Okay, so if we do Control Enter, we'll get a plot down there in that bottom pane with, as I predicted, points at 1, 1, 2, 2, 3, 3, etc., etc. And so we can resize this. So at the moment, that's not a very, it's not a very. This is supposed to be a square, really, because we've drawn 10 by 10 here. So let's just change the size of that pane. So I can maximize it by pressing this button here in the top right hand corner of the pane, and that will make it take up all of this half of the screen. So up it goes, and the plot will dynamically resize and will be shown in the whole pane. So that looks a bit more like it. This is not the best plot in the world, but it was a single line of code, about 10 characters, 15 characters to produce a plot in this window. If I tried to do that at the command line in the console in R, that would be much more problematic. You could also make your plot window take up your entire RStudio window, so you can zoom in on all of these different panes and they will take up the whole window, so we can zoom plots, and our plot will become the size of our RStudio window, and again will resize itself dynamically to the size of the canvas that it has available to draw on. Okay, so I think at this point I'm gonna pause and say, well, I think we've probably done enough for today. So we've installed R and RStudio, and we've done a little bit of testing around. I fully anticipate that there will probably be some issues with getting R and RStudio up and running on some people's machines. What I would suggest that you do if you're having issues is leave a comment on the video once the archive video is up. Leave a comment on the video rather than in the live comments, and then those problems hopefully can be solved with and the comments will be an artifact with a lasting solution to each of the problems that crops up. Okay, so if you're struggling to get properly going with R, then leave a comment and I'll endeavour to get solutions to those problems as quickly as possible, so that people can follow along over the next few days as I begin to introduce more aspects of R. Okay, so there we go. That's our beginnings with R. We will do much more, obviously, over the next few days. I'm going to introduce a whole load of concepts in R that will help us get going in future lessons. And then we'll turn our attention to bioconductor and to RNA-Seq analysis in a week or maybe 10 days. Okay, so that's it from me for today. Thank you again to everybody for dropping in live and tuning in. Thanks to everybody watching on catch up, and I hope to see you all tomorrow. Stay safe, keep well, everybody, and see you tomorrow. Bye.